Welcome back to MicroVerse. In the previous lecture, we'll look at the shape and arrangement of a bacteria. But an important question remains: what gives a bacterial cell its characteristic shape and structural strength? The answer is bacterial cell wall. So today's question is: what is the bacterial cell wall and why it is so important? So the bacterial cell wall is a relatively rigid structure located external to the cytoplasmic membrane. So its major structural component in most bacteria is peptidoglycan. The cell wall provides mechanical strength, maintain the characteristic shape of the bacterium, and protect the cell against the osmotic lysis. There is, however, an important exception: mycoplasma. Mycoplasma lacks a cell wall. This is clinically important because its membrane contains sterols, which helps provide the stability. So, where is the cell wall located? So, let's visualize the basic organization of a bacterial cell. From outside towards the interior, we can broadly think of cell wall, then cytoplasmic membrane, and then cytoplasm. The exact envelope structure differs between the bacterial groups, and we will discuss that in detail in our next lecture. So, for today's lecture, focus on the central concept. The cell wall is the rigid structural layer outside the bacterial cytoplasmic membrane. So, what is peptidoglycan? Now we come to the most important component, which is peptidoglycan. Peptidoglycan is a unique bacterial polymer made up of alternating sugars molecule, N-acetylglucosamine and N-acetylmuramic acid. The sugar chains are connected to short peptide chains. The peptide chains cross-link adjacent glycan strand and producing a strong mesh-like structure. This cross-link network is what gives the bacterial cell wall its mechanical strength. So remember, peptidoglycan is made up of sugar plus peptides and form cross-link. Then why is the cell wall important? The first major function is maintaining cell shape. The rigid wall helps bacteria maintain their characteristic morphology. Without appropriate structural support, the cell cannot maintain its normal form. Then second one is a protection against osmotic lysis. So this is the one of the most important function. Bacteria may exist in environments where water tends to move into the cell. The cytoplasmic membrane alone cannot withstand the resulting internal pressure. The cell wall provides mechanical resistance and prevents the cell from the bursting. Therefore, cell wall provide protection against the osmotic lysis. This is also why bacteria without a cell wall behave differently from typical walled bacteria. Then third one is mechanical protection. The cell wall provides structural strength and help the bacteria withstand mechanical stress from its environment. It is essential acts as a protective framework around the cell. Then fourth, role in the bacterial growth and division. The cell wall is not simply static structure. During bacterial growth and cell division, the cell must be continuously synthesized and remodeled. This makes cell wall synthesis an important target for several antimicrobial agents. Then we see the clinical significance. This brings us to an important connection with the pharmacology. Several important antibiotics interfere with bacterial cell wall synthesis. For example, beta lactam antibiotics such as penicillin, cephalosporin interfere with the enzyme involved in peptidoglycan cross-linking. Because human cells do not possess peptidoglycan cell wall, this provides an important basic for selective toxicity. So, the bacterial cell wall is not only the important for understanding morphology, it is also highly relevant to antimicrobial therapy. So before we finish, remember the classic exception, which is mycoplasma. Mycoplasma does not possess a conventional bacterial cell. Therefore, antibiotics that specifically target peptidoglycan synthesis are ineffective against the mycoplasma. This is very useful clinical correlation. Then if I ask what is the major structural component of the bacterial cell wall, answer is peptidoglycan. Then what is the function of peptidoglycan? Which gives shape, strength and protection from osmotic lysis. And then remember, mycoplasma is cell wall deficient. Remember the three words shape, strength, and survival. So, shape maintain bacterial morphology, strength provides structural rigidity, and survival means prevents osmotic lysis. So, today's question goes what is the bacterial cell wall? It is rigid structural layer outside the cytoplasmic membrane with the peptidoglycan as its major structural component in most bacteria. It maintains bacterial shape, provides mechanical strength, protects against osmotic lysis, and serves as an important target for antimicrobial therapy. But there is an important difference between the bacterial groups. Is the cell wall of gram-positive bacterium is the same as that of a gram-negative bacterium? Not at all. In the next lecture, we will compare their structure and understand why are gram-positive and gram-negative cell walls are different. So thank you for the learning with MicroVerse. Let's master microbiology, one concept at a time.